Hey everyone, and welcome to today's Explainer. Today, we are settling in for what I think is a truly fascinating lecture on the microscopic machinery of life. We're talking about enzymes. If you've ever stopped to wonder how on earth our bodies manage to digest food, or heal a paper cut, or honestly, even just allow us to take a breath, well, the secret is all in these incredible biological catalysts. It's a wildly cool topic, and I'm super excited to explore this material with you. We're going to use some great diagrams to really visualize these concepts as we go, so let's just get right into it. Here is a quick look at our syllabus for today's journey. First up, we'll meet these biological catalysts. Then, we'll break down the actual anatomy of an enzyme, followed by exactly how they go about catalyzing reactions. After that, we'll run through the 6 major enzyme classes, take a look at the optimal conditions they need to thrive, and finally, we'll wrap up with their everyday functions in our world. All right, let's dive right into Section 1, meeting the biological catalysts. To kick things off, we really need a core definition. What are they? Well, enzymes are biological polymers, and the vast majority of them are proteins. And their one main job? To catalyze or significantly accelerate biochemical reactions. Just think about this for a second. The human body is this insanely complex system of tissues and organs, right? It requires near instantaneous chemical reactions just to keep functioning. Without these microscopic chemical machines speeding up processes like respiration and digestion, sustaining a healthy life literally wouldn't be possible. The initial stage of almost every single metabolic process in your body depends entirely on them. So the big question is, how this transformation actually happens. If we map out a simple linear flow, metabolic processes always begin with a specific starting molecule, which we call a substrate. The enzyme acts on this substrate, and through that specialized enzyme action, it converts it into a totally distinct molecule, the product. It's basically an elegant microscopic assembly line happening millions of times a second. Let's see how this physical structure is actually built in Section 2, Anatomy of an Enzyme. At their absolute core, enzymes are just linear chains of amino acids that fold into highly specific, incredibly complex three-dimensional structures. And it's that exact sequence of amino acids that dictates the structure, which then totally dictates the enzyme's specific job. Now, enzymes can be absolutely massive compared to their substrates. To put it in perspective, they range from just 62 amino acid residues all the way up to a whopping 2,500 residues, like the ones you'd find in fatty acid synthase. But here is the craziest part. Only a tiny, hollowed-out little section of this massive structure actually does the heavy lifting in the chemical reaction. That little pocket is called the active site, and it's made up of a catalytic site and a binding site. But, some enzymes can't do the whole job entirely on their own. They need a little help. They rely on these non-protein wingmen called cofactors. And when an enzyme pairs up with its cofactor, it forms a complete, ready-to-go unit called a holoenzyme. There are 3 main types of these helpers. First, you've got prosthetic groups, like the organic molecule FAD, which are tightly permanently bound to the enzyme. Then you have coenzymes, like the carrier NAD, which only attach when the actual catalysis is happening. And finally, metal ions, like zinc, which are required right there at the active site to form coordinate bonds. So, now that we know what they look like, let's move to Section 3 and see how they actually catalyze these reactions. Okay. For any chemical reaction to happen, molecules have to collide with just the right orientation and enough energy to actually react. Think of this energy barrier like a massive hurdle on a track known as activation energy. Without an enzyme, a biological reaction might require such a huge leap of activation energy that it would happen way too slowly to keep you alive. Enzymes step in and function by providing a specialized surface that lowers this exact barrier. They basically shrink the hurdle, allowing the reaction to zip toward equilibrium significantly faster than it ever could alone. Which naturally brings up the ultimate question for this part of the lecture. How does this catalytic cycle actually work down at the molecular level? Let's break this mechanism down step by step on our virtual whiteboard. In step 1, the free enzyme, we'll call it E, and the substrate, S, combine at that active site we talked about earlier. This forms a temporary intermediate transition state called the ES complex. By making and breaking chemical bonds right here, the magic happens. Then, in step 2, this complex disintegrates. It spits out the brand new product, P. But here is the absolute most important takeaway. The enzyme, E, is released entirely unchanged. It's identical in quantity, identical in chemical properties, and completely ready to grab a new substrate and do the whole shebang all over again. Now, historically, scientists thought of this binding like a rigid lock and key.
The idea was that the substrate fit perfectly into a hard, unyielding active site. But the much more accurate modern understanding is something called the induced-fit model. In this model, the initial interaction is actually kind of weak, but those weak interactions quickly induce a conformational change. Literally, the enzyme shifts its shape, like it's giving the substrate a hug, to strengthen the binding and bring all those catalytic sites perfectly into alignment. Moving right along, let's hit section 4, the 6 enzyme classes. So, enzymes are very strictly categorized by the specific type of reaction they catalyze, thanks to the International Union of Biochemists. We can basically map out 6 functional classes. First, oxidoreductases, which handle oxidation and reduction. Then, transferases, and true to their name, they transport chemical groups between molecules. Hydrolases, like your digestive enzyme pepsin, use water to cleave bonds. Lyases, like aldolase, also break bonds, but without using water. Isomerases catalyze structural shifts to change a molecule's physical shape. And finally, ligases, like DNA ligase, which essentially act as biological glue to join 2 molecules together. Now, these are highly sensitive microscopic machines. So let's talk about section 5, the optimal conditions they need to survive. 37. That is 37 degrees Celsius. That's standard human body temperature, and it sits right in the absolute sweet spot for human enzymes, which operate best right between 35 and 40 degrees. This specific temperature allows for the perfect number of molecular collisions without completely melting the machinery. Now, what I find really fascinating about this is how going outside that optimal temperature halts enzyme activity, but for totally different reasons depending on which way you go. Extreme heat actually pumps in so much energy that it completely disrupts the weak hydrogen and ionic bonds inside the enzyme. The protein denatures, it totally loses its 3D shape and therefore its function. On the flip side, extreme cold doesn't denature the enzyme at all. Instead, it makes the molecules move so incredibly sluggishly that the collisions between the enzyme and the substrate just drop off a cliff, which effectively hits pause on the whole reaction. And hey, it's not just temperature. pH levels are absolutely vital. Most enzymes prefer a nice neutral pH between 5 and 7. Substrate concentration matters too. If every single enzyme is currently busy, you hit a saturation point, meaning tossing in more substrate won't speed anything up. Then there's salinity. High salt levels disrupt the attractions between charged amino acids, denaturing the protein. This is exactly why the Dead Sea is, well, dead. The insane salt levels create an environment where enzymes just cannot function. Finally, inhibitors. These are tricky chemicals that mimic substrates and actually compete for that active site, blocking enzyme activity. We actually use this exact mechanism intentionally in medicine to fight off bacterial infections by inhibiting the bacteria's vital enzymes. Pretty clever, right? All right, let's bring all this complex biochemistry back down to earth in section 6, everyday enzyme functions. Because truthfully, enzymes are everywhere. Inside you, right this very second. Protein kinases are facilitating signal transduction, while ATP synthase is working nonstop to generate your body's energy. Digestive enzymes are busy breaking down your last meal into smaller, usable substances. But they also totally shape our food and culture. The simple act of baking a loaf of bread, that relies on fermentation, specifically the enzyme action in yeast breaking down sugar to create carbon dioxide, which is what puffs the bread up. The fermentation of grapes or hops to make wine and beer, all driven by specific enzymes. They are quietly running the entire show, regulating cellular activities, and literally keeping the biological world turning. Which brings us to the end of today's explainer. But before we go, I want to leave you with a bit of a provocative thought to chew on. If nearly every critical life process, from the baseline energy in your cells to the grand structure of our ecosystems, relies on these incredibly precise yet totally chemically fragile biological catalysts, how different would our world be if they just stopped working for one single second? Thank you so much for joining me for this lecture. I hope these diagrams and our chat have really illuminated the microscopic machinery that makes your life possible. Keep asking questions and stay curious.